Well, hello there, AP Psychology students. Today we're going to be talking about one of my favorite topics. Oh, wait, I say that all the time. Oh, well, it's just true because there's a lot of things I'm amazed about. And one of the things I am most amazed about with the human body and mind is our vision. We're going to be looking at the components of the eye, which you should have already started to look at, as well as some theories about why or how we see vision. And tomorrow we'll get into perception. I do think vision is just so uh, remarkable, and the eye is so intricate and beautifully made. It is mind-boggling to me. You already have looked at the structures of the eye on your um, diagram. And so if you have not recorded these yet, I just recommend that you pause the video and you fill out um, your diagram with these structures. I'm going to point out a couple of things, however, that need special attention. First of all, the lens is a rather what people have dissected sheep eyes tell me is it's kind of thick and firm, whereas the vitreous gel in here is very liquid, kind of like a soft jello. It's so cool because it actually learns to become thicker as we're looking at near objects and to thin out as we're looking in the distance. So some people, maybe some of you are like this. You were nearsighted when you were younger and didn't know it, and so you would go like this, and you would try to sort of manually thin out the lens because it wasn't focusing correctly. And that's kind of what happens. If you have glasses and you take them off and you go like this, you might be able to get yourself to see completely clearly. The other thing that I want to point out are the rods and the cones. So you can see down here, the cones are fat. They are what sees the color, or they're what receives the color. The rods, it's quite obvious in special testing that the rods are specialized to notice periphery, things going on the side, and motion. And so you can start to identify movement way off in your peripheral vision before you can start to see color. And if you were in class, we would do a demonstration and I would show you this fact, but it is very interesting how these two specialized cells in the retina help us to see color or periphery in motion. Now, on its way to the visual cortex, light comes through, and the retina's in the back of the eye, light comes through and it excites these little rods and cones down here. It's a photochemical reaction, and this is where the actual point of transduction occurs. It occurs in the retina and more specifically in the rods and the cones. So these are stimulated, and you can see there's a little synapse here, and these would be the dendrites of the receiving cell. Here's the cell body. The rods and cones transmit a message to the bipolar cells. As the bipolar cells um, send their message, they move on to a special type of cell called the ganglion cells. Now we have fewer and fewer cells as we go through this system. We've got about 5 million rods and cones in each eye, and then the number of cells narrows down. Now what happens to the ganglion cells is that the axons of the ganglion cells form what is called the optic nerve. So this is the nerve, it's a bundle of axons from the ganglion cells. Now, as you know, your part of your visual field goes to your right side of your brain, part of your visual field goes to the left side of the brain, and the place where the optic nerve separates is at a place really not very far back behind the front of our head called the optic chiasm. That is this structure right in here. In this area, the cells will divide in half and they will send the information then through the thalamus and on to the visual cortex, which is back here in the occipital lobe. So I just want you to know that order of events, and and then from the visual cortex, that information is first received, and then it goes through multiple parts of the brain for identification. Some vision processing concepts. Well, I pretty much obscured the input image here. It is actually a very colorful bird. But what happens when we first see something, and this is just so split second fast, again, amazing. It's the word I use a lot, is that these feature detectors in our occipital lobe actually detect the lines and the edges. And parallel processing also occurs in the occipital lobe. So color, movement, form, and depth are all received simultaneously in the occipital lobe. Now, as we've been talking about, the occipital lobe, if it is damaged or part of it's damaged, you might have hallucinations, or you might have even brain blindness. All right, let's look at color vision. This is something that's so fascinating. So if you think about it, if a tree falls in a forest, does it make a sound? Well, it does depend on something or someone with ears to hear the sound, but the sound waves still exist. But what does not exist in nature is color. It does not exist. Color is purely a construct of our eyes and our brains. And every single living thing experiences color differently. And it is, for humans, something that we only perceive from 40 to 70 nanometers in the electromagnetic spectrum. That is really fascinating. There are some animals like bats that can register radar, but for humans, it's just this little band of this spectrum that goes into our eyes and then into our brain and allows us to see the colors that we see. They don't exist in nature. There are no brown rocks on Lake Superior. They're only brown because we see them as brown. Some people have asked that age-old question, is your green the same as my green? Is your blue the same as my blue? Well, we really have no way of knowing. It's possible that we do see them slightly different because of our eyes and because of certain structures in our eyes. So where does our color vision come from? The first theory out there is the trichromatic theory of color vision. This was also known as the Young-Helmholtz theory. I don't think that name will come up. You'll probably hear it as trichromatic theory, but some people still do use the Young-Helmholtz theory. So according to this theory, we have three types of cones. Remember those fat structures of those cells at the back of the retina. And these are red, green, and blue-violet. Now, you combine them together, and these form up to 7 million colors. It's believed that humans can see up to 7 million colors. Now, what's really interesting, and I listened to a podcast on this recently, there are actually some humans have 4 cones. They have like a cone for orange, and they actually can see more colors. They're called super seers. And there are some tests that can be done to see if you are a super seer. If you're someone that can see more colors than the average person, this can only be discovered through specialized testing, and it is probably something that you inherited. So color blindness is thought to be specifically an issue with the eyes themselves, with the retinas and the cones. Now, this is a common type of test that you might be given to see if you're colorblind. And in the top, you should see the number 7-4. Okay? If you can't, and let's hope this isn't the first time that you've looked at one of these, but it's possible that you have dichromatic color blindness. This tends to run in families. It's definitely more common among males. When I ask students in my class about color blindness, almost everyone knows someone who has color blindness. Now, it does vary in degree. The bottom circle, I think, is a little harder to see, and it has 45 in it. Again, I know my picture's obstructing part of it, but what is the cause of dichromatic color blindness is a lack of the function of red or green cones. Now, I know someone who is very colorblind and cannot distinguish red and green at all and would not be able to see any difference between those colors in the top circle. But color blindness varies a great deal um, in its intensity and how it is manifested. Red-green color blindness is the most common. There are some people that have color blindness with some blues as well. Monochromatic color blindness is a result of having no color receptors. In my lifetime as a teacher and asking students about color blindness, one time in my life someone said that they know someone without any color receptors. So we tend to take color for granted. We love color. Color brings a lot of vividness in our lives. But for someone who is born with dichromatic or monochromatic color blindness, they don't know anything differently. And you've maybe heard the expression, you can't miss what you don't know. And that is true. They can still see detail and edges. And I wouldn't say that it has a particularly bad outcome for them unless they maybe are into something like fashion design or interior design. But otherwise, most people can find ways to work around it. This is an example of a painting here with no color blindness, and then some various types of color blindness. So I want to tell you about the mantis shrimp. Okay, so this little dude here, he is amazing. Like, amazing. Wait, I've already said that today. The mantis shrimp has 12 types of photoreceptors. And look, it even says here, they have such amazing eyesight compared to other crustaceans. So most of them, many of them live in coral reefs and in areas where being able to differentiate a large number of colors probably gave them an evolutionary advantage. They can see both circular and linear polarized light, hyperspectral color. This is different from what humans see. Now, we can't really quite understand what that looks like to the mantis shrimp because our eyes aren't capable of it, but it is something that researchers are well aware of, and I just think it's an interesting side note. Now, I'd like you to actually pause your camera for 30 seconds and stare at the blue star in the center of this picture. So just pause and stare. Okay, when you're ready, make sure you've stared at it for 30 seconds. Take a look at this picture. You should have seen the picture with a pretty dramatic color, natural color of grass and sky, and then it very quickly changes to just a black and white image. Now, if you want to do it again, you can pause it and try it again. And if you didn't see it the first time, pause the video for a little bit longer and stare at the blue star. There we go. Now, this is a very textbook version of the phenomenon that we're looking at here. Fix your eyes on the white circle in the center of the flag. Now again, pause the video and stare at it for about 30 seconds. Okay, so if you haven't paused the video, pause it now and look at the circle. All right, I'm going to change the slide. This is a fun one. Most of you should be seeing a red, white, and blue flag. If you didn't, I would suggest that you go back, pause the video, and look at this for about 30 seconds. All right, red, white, and blue flag. This is an after image. There's nothing on this slide. It is completely blank. So why does this happen? This happens because of something called the opponent process theory. So in the opponent process theory, this describes how somewhere in a visual system, not in the rods and cones, and we don't know exactly where, but somewhere in the visual system, there are three pairs of opposing receptors, red and green, blue and yellow, black and white. And when we look at, for example, the green, yellow, and white or black for a long time, those receptors get tired, they get fatigued. And so then when we look at something else, a blank screen, for example, then we're going to see the opposing colors, in this case with the flag, the red, white, and blue. So this is really interesting as well. So both the trichromatic theory and the opponent process theory are important ways of understanding color vision. Now, if you look at the object that I have on the screen, some of you might see motion, very slow motion. This is called anomalous motion, and it's not really well understood, but it is being studied by visual researchers and psychophysicists. There are several of these. I might put one on our Schoology website of these. They can kind of bend your mind for a little bit, but you might see some motion in that picture. This one, I for sure see motion. If I look at the center, not only does the center appear to be moving clockwise, but the two circles on either side are moving in opposite motion. So again, this is an example of anomalous motion. There is no motion, but our eyes and our brain or somewhere is wired to see motion. How do we actually see motion has been of interest, though, to vision researchers. And so our brains are actually wired to analyze biological motion. So we see motion through still frames like you see in this picture. And I know it's kind of a weird picture of someone doing some acrobatics, but nevertheless, it does illustrate, it's believed, how we actually see motion in small, very quick sequences of actions. And then the phi phenomenon is something where we see when we see still images in succession, this allows us to perceive motion. So I'm going to put an example of the phi phenomenon on a separate link on Schoology. And this is it for today. Make sure to look at your ABA and have a great day. Thank you.